Oh, okay. All right. I put in that maybe if we met on Tuesday before you took the exam that's available Monday and Tuesday, then that would be a good thing. So I will be at the airport, and even all day Tuesday will be bad for me. So I'm thinking Monday, even if some of you can just be there and I recorded if there's any extra stuff. So I'm not going to be worried about an actual, attendance for next week's meeting, being that I can't hardly even be there myself. But that's the only week that's going to be like that because my daughter has to move to North Carolina. So if we want to meet on Monday, I should be fine because I'm going to be 3 hours later than you guys. I should be fine. Let's see, what time is our class meeting? It would be at 9 on Monday. Noon. Later in the afternoon would be better because we're just going to be unloading on Monday. So be thinking while we go through the class meeting right now, and then we'll throw up in the chat what kind of ideas, what people want to do. So be thinking what we need to accomplish for next week. Okay. Let me just check a couple more people. I got everybody else. Gabby, did I get you? Yeah, I did. Okay, so with our class meetings, what I intended is to go over the most important things out of each one of the chapters or the set of lecture notes that really need some reinforcement, I think. And then any questions that you have, anything that you guys want me to clarify, I guess that's what the intent of meeting online at least once a week is. So stop me along the way if you have any questions. To unmute yourself, you just hit the space bar, or you can unclick to mute yourself. So what I'm going to do is just go through my PowerPoint that I have for the endocrine system. I want to do that for about a half hour, and then the 40 minutes, and then the second half we'll hit some hematology stuff and talk about blood clotting, because that's usually the rest of the stuff is, just kind of memorization. What are the different types of blood cells for? And but the clotting process is a thing that can be a little more complicated. So I'm going right to the to my PowerPoint on the endocrine system, and make that big in a minute. Okay, and then make us small. All right. When you go into the lecture notes and into the PowerPoints and you watch my videos and things like that, I always have objectives. I know I wrote them really tiny because I didn't want them to be on 2 slides. So I want to do that right now and see if anybody has any particular questions about the objectives, and give you an idea of which ones are really more important to remember, the concepts that take maybe a little bit more to effort to understand and learn. All right, purpose of the endocrine system, super important. It is the other homeostasis maintenance system that we have in our body with the nervous system. That's going to be our immediate short-term homeostasis. Just, I got to jump out of the way of a car, or I've got to not touch that hot stove or something like that. But the endocrine system is more long-term homeostasis to help us keep our body environment more within a normal range, or some outside external stimuli. But I find with most of the endocrine, with the hormones and processes that go on, it's really trying to maintain your internal environment. And so we'll look real quickly at how does the endocrine system and the nervous system regulate homeostasis, and then we'll look at, well, what really does the endocrine system govern in our body? So the processes that are controlled. Exocrine endocrine glands, I'm going to hit that real quick right now. Remember, with an exocrine gland, there is a duct, and so the secretions like the saliva and pancreatic juice and bile and sweat, all of those secretions will be secreted from their glands through a duct into a cavity or onto the surface of your body. On the other hand, with an endocrine gland, the secretions will go right directly into the bloodstream. So you have an extensive capillary network in all of the endocrine glands, so that the hormones will just automatically go right into the capillaries. And the capillaries tend to be quite porous that we have within endocrine glands. Fenestrated capillaries that we'll talk about with the blood vessels in the next unit. So because the hormones can have various sizes, some are big, some are small, and that way all of the hormones are able to get into the bloodstream. Hormones classified chemically, they come in two kinds. They come in protein hormones, and they come in steroid hormones. But I'm going to look specifically at that. When I look at how do the hormones bring about the effects on their target tissues, and then we want to look at hormone release regulation. There are three ways that hormones can be stimulated to be released, and then we'll look at how well do hormones, what determines how well hormones affect their target cells, and when hormones act with one another, interactions with one another, what kinds of interactions are those? We need to look at the relationship between the hypothalamus and the pituitary gland. Remember from the nervous system that the hypothalamus is control of the autonomic nervous system, which really is maintaining homeostasis within your body, heart rate, breathing rate, all of that kind of stuff that you don't have to think about. And part of the way that the hypothalamus is going to govern that is through influencing the pituitary gland. So that's an important thing we need to look at. Then we'll look at the different hormones. So the hormones that come from the adenohypophysis, which is the anterior pituitary, the neurohypophysis, the posterior pituitary, and then thyroid hormones, parathyroid, adrenal gland hormones, pancreatic hormones, and then the last one. So that shouldn't take us long to get through. In the lab portion of the course, so if you've been to that page that says the quiz terms for quiz 1 and the lab practical, you'll want to be familiar with this picture and the location of each one of the glands. So pineal gland, hypothalamus, thalamus, pituitary, so on. So make sure that you're familiar where the glands are found within the body. So we talked about hormones and the control system. So if you review through this, I'm not going to spend a lot of time on it, but you know the nervous system is very, very rapid and short-lived because the neurotransmitters are just immediately removed from the synapse, whereas the endocrine system is longer lasting because the hormones are in the bloodstream. And so as they travel through the bloodstream, anywhere that we have a target tissue that has a receptor for that hormone, then that target tissue will be influenced by that hormone as long as the hormone is there. So hormones need to be removed. Like, so in this last bullet point, hormones last longer in the blood. The response of the target tissues are more prolonged. So we say that hormones have half-lives, which is how long does it take either the kidney or the liver to remove that hormone from circulation. Some hormones have really long half-lives. Thyroid hormone, cortisol, estrogen, testosterone. Some of those last longer, but immediate hormones, you're all familiar with the fact that when you slam on your brakes and your heart is beating 100 miles an hour, it doesn't immediately stop right after that because you still have the epinephrine that was secreted from the adrenal medulla running through your bloodstream, and then that has to be removed by the kidney and the liver. So fortunately, it has a very, very short half-life, doesn't stay in your bloodstream very long, so that heart pounding feeling doesn't persist for hours at a time, which I would hate that. Okay, so what does the endocrine system govern? So the hormones that are secreted, what are their 5 jobs? Okay, so the first one is reproduction. And none of these processes would happen without hormones. So reproduction would be FSH, LH, estrogen, testosterone, progesterone, GnRH, and all of those have some effect on the actual production of the gametes or the reproductive tracts that we have in male and female. Growth and development, growth hormone, some cortisol, some, those are the main more anabolic, well, growth hormone is definitely an anabolic hormone. Oh, testosterone. Just trying to think. I know there's another one. Mobilizing body defenses against stressors. So this one usually gives my students a little bit of pause, like, what does that really mean? So we have to think, well, when our body is stressed, what gets released? So there's two kinds of stress that we have in our body. We have chronic stress and we have acute stress. And in fact, I have a slide about that, so I think I'll talk about it then. So that's what we're looking at. How do we handle long-term stress? How do we handle short-term stress? Maintaining electrolyte, fluid, and nutrient balance in the blood. Probably the most common of the functions as far as numbers of hormones that we have. So we have aldosterone and ADH and insulin and glucagon to help regulate those substances within our bodies. And then cellular metabolism and energy balance is going to be thyroxine to help make sure that we are burning our sugars like we're supposed to, our energy. Okay, so there's the definition about exocrine and endocrine glands, so you can look at that again if you have any questions. All right, so here are the two types of hormones, the two classes of hormones. So amino acid based, we can call them polypeptide hormones or protein hormones. A majority of the hormones are made out of amino acids, and they vary in size from just a few amino acids, 6 or 9 amino acids, up to 100 or more. So simple amino acids to very large proteins. Steroid hormones, on the other hand, are made from lipids, or lipid category. So cholesterol is the base molecule, and then we're going to modify the cholesterol to produce aldosterone, cortisol, testosterone, estrogen, and progesterone. So all of those hormones are going to be steroid hormones, and there was that. We also have thyroxine, is similar to a steroid. It is not a steroid hormone, but it acts like a steroid hormone, and that'll make sense here in just a minute. All right, so how do hormones, what kinds of things or processes that happen at the target cells when stimulated by their hormone? So here's the key. So you can't tell a cell to do something different than what it's going to do. So you can't tell a chondrocyte, so a cartilage cell, you can't tell it to secrete sweat. It just, whatever that cell is programmed to do, that the hormone is going to tell it do more of it or do less of that activity. Okay, so that's why we have a variety of different cells in our body to carry out different jobs, different functions, and so we have different hormones that act on different target tissues to get the different processes done in our body. But you can't, all that the hormone is going to do is tell the cell do more of that or do less of that. So what are those things that they do? All right, so either they alter membrane permeability or membrane potential by opening, closing ion channels. Okay, really good example, and we'll look at the hormone chart before we leave today, but a really good example of that is epinephrine. So, and norepinephrine. Why does our heart beat faster? Well, it's because it depolarizes more rapidly. So the cell becomes more permeable to sodium so that the heart can beat faster. In B, stimulates the synthesis of proteins, hormones, or other certain regulatory molecules within a cell. So this word right here, tropic hormone, it's not tropic. I don't care how you pronounce it, but if you hear the word, it's a tropic hormone. So these are hormones that are going to stimulate other hormones. And I have a slide for that that'll show you what tropic means in just a minute. So we'll come back to B. Okay, C, activates or deactivates enzymes. Let's take glucagon, for example. So glucagon is a hormone that's released by the pancreas, the alpha cells of the pancreas, when our blood sugar is low. And the quickest place where we can raise or get some glucose from to raise our blood sugar level is our liver. Because when we eat our food and we digest it down into the monomers of glucose monosaccharides, that some of the glucose, instead of just going to our brain and our muscles and our adipose, some of that glucose is going to go to our liver, and it's going to get converted to glycogen, which is the carbohydrate storage molecule within our bodies. We store it in our liver, we store it in our muscle, and there it'll stay. And we can store a little bit of glycogen, not a lot. It's not going to save us over days and days and days. That's when we start using up our body fat, but it'll supply our needs for a few hours. So when we've been asleep all night, for example, and our blood sugar is low, we get up in the morning, our pancreas is gonna detect that there's not very much glucose in our bloodstream. It'll secrete glucagon. So glucagon will go to the liver, and it will activate the enzymes that cause a process called glycogenolysis. And so what glycogenolysis does is break up the glycogen molecule into its component glucose molecules and then secrete those out into the bloodstream. So there's an example of activating enzymes. D is also associated with tropic hormones, because not only were the tropic hormones telling the gland to make the hormone, but then it tells it to secrete it as well. So an example of this would be FSH, follicle stimulating hormone, in males and females will, well, let's do it in a female because it has a different job in a male. So FSH in the female is going to stimulate cells within the ovary to produce estrogen and then secrete estrogen. Males have another hormone called interstitial cell stimulating hormone. Used to be called luteinizing hormone in a boy, still called LH in a girl, but that ICSH or LH is going to stimulate the testes, the interstitial cells, to release the testosterone. So that's what tropic hormones are going to do. And then stimulate mitosis, you're probably familiar with growth hormone. And actually growth hormone is going to go, growth hormone is more of a metabolic hormone, increasing blood glucose levels, but in order to get it to cause growth, growth hormone is going to go to your liver, and it's going to stimulate somatomedin or IGF-1, so insulin like growth factor 1, and then that goes to our bones, goes to the epiphyseal plate and stimulates mitosis of the chondroblasts in your epiphyseal plate. Okay, so there was a whole bunch of stuff on that. Sorry, you guys. So here's how a tropic hormone works. So the hypothalamus needs to stimulate the anterior pituitary. So this is a gland, try to get hormone to be released from the anterior pituitary. So it's going to send a releasing hormone here, thyrotropin releasing hormone, or TRH, and that will stimulate cells within the anterior pituitary to then in turn release TSH, thyroid stimulating hormone. So these two hormones are tropic because they're acting on glands. So TSH is going to act on the thyroid gland, and then we'll ultimately get the thyroid hormones, T3 and T4, that we want. And then they'll go to their target cells and do their job. But TRH and TSH are tropic because they are stimulating these two glands to get them to do their job. So you can see stimulate, 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 and then we have regulation of this secretion by a negative feedback mechanism. So this homeostasis, so that we're not constantly making thyroid hormone, then the thyroid hormones themselves, T3 and T4, are going to go back here and they're going to inhibit anterior pituitary secretion of TSH, and they're going to go back here and inhibit hypothalamic secretion of TRH. And so most of our hormone mechanisms, negative feedback mechanisms, work just like this one. So the thing that we make in the end will go back and stop the production until these levels drop really low, and then we can start all over again. All right, now second messengers. I'm going to show you a picture of how second messengers work. Most of the hormones that we have, we said, were protein hormones. And unfortunately, protein hormones cannot get into the cell to tell the cell to do its job. So inside of the cell, we have nuclei, we have chemical cascades, enzyme cascade processes that stimulate the cell to do its job. But they're on the inside, the hormones on the outside. So how do we let the target cell know it's time to do your job, or increase the rate at which you're producing some product or something like that? So, so this is called the second messenger system right here. So in this first case, the hormone is the first messenger. It's going to bind to a cellular receptor. So it's going to be an integral protein. It's got a receptor site on the outside, and then it's got a portion on the inside that can activate a process here, or a protein on the inside. So we have this protein right here called G protein. Now, once G protein becomes active, then it's going to go over here and stimulate this enzyme within the cell membrane called adenylate cyclase. I don't need you to know that. We just need to know that binding of the hormone to the receptor will activate G protein, and then G protein will stimulate the production of this molecule right here called cyclic AMP. Now, cyclic AMP is our second messenger. So if we take a look, let me draw. I forgot I can draw with this. Okay, so here we're going to go in and we're going to bind, and then we're going to activate, and then we're going to go this way, and then we're going to make this guy. So this is the one that we want, because cyclic AMP as our second messenger is going to turn on this protein kinase right here. So it's just sitting around in the cell, not doing anything. And then once the levels of cyclic AMP rise, then the protein kinase becomes active, and then we get a variety of cell processes, whatever the cell needs to do. We have that process described right here. So I'm not going to read that because you can read it on your own, but I just showed you that picture. All right, there's a second messenger system, another second messenger system, I guess I should say, that uses calcium as the second messenger. Actually, it's kind of like the third messenger. So let me show you how that works. Okay. All right, so again, the hormone is going to bind to the receptor. Again, we're activating G protein. But in this case, we have phospholipase C, and then diacylglycerol, that's going to be here in the cell membrane. So what we're doing is activating these chemicals inside of here. And then we're going to produce this as our second messenger is IP3. So we converted PIP2 to IP3. This is our second messenger, but what's really going to do our job is an increase in the intracellular levels of calcium. So remember back when you were doing muscles and you had the sarcoplasmic reticulum that stored the calcium for muscle contraction. We've got the calcium coming out, and then we also have this calcium because it can trigger cell responses, or we complex it with calmodulin, and then we can get the processes going on within the cell. Like with epinephrine, when it causes your, because it's a catecholamine, when you're causing your heart to beat harder when you're in sympathetic stimulation. That's its job. So either way, whether we use cyclic AMP or we have this IP3 to increase our intracellular calcium, that's going to be the triggering mechanism. Okay, so we've got to increase our levels of IP3 and calcium in order to carry out our job. So those are the two second messenger systems that we have. So depending on what hormones, so here we can see catecholamines, TRH, ADH, LHRH, oxytocin, or where's my little thingies? Or if we go back to the go back to this one, most of them are going to use, most of the protein hormones will use this one to raise intracellular levels of cyclic AMP. Okay, so if you're a protein hormone, let's just go back, oops, back to this side. If you're a protein hormone, you need the second messenger because either you're too large to fit through the cell or you're too polar to fit through to go through the phospholipid bilayer. You can't get in. And we won't watch that video. Okay, so what if you're not a protein hormone? Then you must be a steroid hormone, excuse me. So how's a steroid hormone going to work? Well, steroid hormones are small. That cholesterol molecule is actually fits between a phospholipid, between two phospholipids. So it's really a tiny molecule. And in addition, it's a lipid. And so it's lipid soluble. So the steroid hormone can slip very easily through the phospholipid bilayer. And all steroid hormone receptors are intracellular. So we've got 3 places they can be. They can be in the cytoplasm, they can be floating around in the nucleus, or they can already be complexed with the DNA. But either way, doesn't matter, you know, because you're not going to have to distinguish between where the different receptors are. You just need to know they're intracellular. So once the hormone comes in through the cell membrane, it has to find its receptor. Because if a cell does not have a receptor for a particular hormone, that hormone cannot elicit its effects on its target cell. So really good example, testosterone. Okay, so let's pretend that this is testosterone and it's going into a muscle cell. So we know that muscle cells have receptors for testosterone on the inside, because why do athletes take extra testosterone? Because they want to build bigger muscles. So we know that testosterone's effect on muscle tissue is anabolic. But what if it were to go to a hair follicle, to the epithelial cells in a hair follicle? Does it make you hairy? No, because there's no receptor for, well, that's not a good example. Because yeah, you do get hairy when you get testosterone. I didn't think about that. I was just thinking rate of hair growth, but you do get hairy. Let's see. Let's take a place, your brain. Gosh, testosterone goes to a lot of places. It does go to your brain. Where does it? It doesn't go to your eyeballs. Okay, so you got that testosterone floating through your body. Can you imagine if you had receptors for testosterone in your eyeballs, that your eyeballs would grow as a boy in puberty? Yeah, no, I don't want that to happen. So if a cell does not have a receptor, then the hormone can be there all at once and it won't trigger an effect. Okay. All right. So let's finish this off. So then once the hormone and its receptor have complexed, then it will go into the nucleus. It will find the receptor binding region on, like a promoter region on a gene, and turn on that gene and get it to transcribe messenger RNA, which will then go out and then translate into a protein. So let's go back to that muscle cell example. So when testosterone comes into a muscle cell, it'll bind to the receptor, they'll go into the nucleus, they'll find the gene for making actin and myosin, the proteins we find in myofibrils. So then we'll make a whole bunch of actin and myosin, and then that will come out, well, the mRNA for actin and myosin. Then we'll make a whole bunch of actin and myosin, and then we'll make a whole bunch of myofibrils. And then that person that's trying to get big and bulky and muscly without doing all the work that you need will then get big and bulky without doing all the work that you need to do that. So that's how the steroid hormones work. Okay, so we said that receptors need to be present on the target cell for them to act on a receptor. What kinds of things will affect how well that hormone will stimulate its target cell? Depends on how much there is in the blood. So a target cell may have an immediate response depending on how much is there, or it may last hours to days. How long will the reaction be there? 20 minutes to hours. Half-life, like with epinephrine, because you just shoot out a whole bunch of epinephrine, so let's get rid of it quickly within a fraction of a minute or 30 minutes when we start to break it down, or whatever it happens to be. Now, here's a really important concept, is that our target cells do not always have the same number of receptors on them. And as a protein, or I mean, as a hormone, is secreted in greater amounts, it will trigger the target cell to increase its number of receptors. So we call that upregulation. So rising blood levels of a hormone may cause more receptors on the target cell. Okay, so that's upregulation, making more receptors for the hormone. However, when we don't want the hormone to be receptive, the target cell to be receptive anymore, then rising blood levels may cause a decrease in a receptor number. And we call that downregulation. And that's going to be depending on the hormone and its function. Do we want more? Do we want less? If we have hormones that are antagonistic to one another, like insulin and glucagon, glucagon's going to stop the cell from making insulin receptors. So it's going to downregulate insulin receptors in our body. All right. Okay, so affinity is just, how strong is the bond between the hormone and the receptor? Stronger the bond, the more the action's going to take place. All right, so now I got to hurry and finish this up. So control of hormone release. So negative and positive feedback. Went through the negative process. Positive feedback is where we just get more and more and more stimulus as more and more hormone is being released, and it doesn't shut it down. There's a couple of examples of that. Prolactin acts on a positive feedback mechanism. So as your baby nurses, more prolactin will get made, which will cause more milk to be made so that your baby can nurse more, so they can grow bigger. And so that's positive feedback. And oxytocin is also a positive feedback mechanism in getting uterine contractions to take place. So as oxytocin starts to be secreted, then it gets some contractions going on in the uterus, which would then go back to the hypothalamus and say, hey, you've got a baby that needs to come out of here. Can you secrete some more? And so then you just keep getting a stronger and stronger reaction until finally the baby's born. All right, so there are 3 types of stimuli. Let's just look at the picture. Pictures are always better. So humoral stimuli is going to be some level in the blood. So calcium levels, sodium levels, potassium levels, water levels, glucose levels, oxygen levels, all of those are things that can change within our bloodstream, and then hormones will be released in response to low levels or high levels, it just depends, in your bloodstream. So that's a humoral stimulus. Some substance in your blood is stimulating the release of a hormone. So in this case, low blood calcium levels will trigger the parathyroid glands to release PTH, which is going to raise blood calcium levels. A neural stimulus, there's only a couple of neural stimuli. One is the only one really that I need you to know is here at the adrenal medulla. So when you're in sympathetic stimulation, you'll get sympathetic neurons that go to the adrenal medulla and tell it to release the epinephrine and norepinephrine. So as soon as you need to slam on your brakes, or as soon as that stimulus is that kicks you into sympathetic stimulation, bam, you get this fire. Oops, where's my thing? You'll get this neuron firing to the medulla, and then you'll get that extra burst of epinephrine and norepinephrine to help you through that acute stress. And then a hormonal stimulus involves the tropic hormones. So here in the hypothalamus, we have the releasing hormones. They're going to go through this hypophyseal portal system that's then going to take the hormone to the anterior pituitary. Then the anterior pituitary cells will produce more tropic hormones, so TSH, ACTH, GnRH, to get these glands to stimulate, to produce their hormones. So hormonal stimulus is a hormone that stimulates the production of another hormone. All right, permissiveness, synergism, and antagonism is how hormones interact with one another. So with permissiveness, you have to have both hormones in order for full function to happen. So we can have reproductive hormones, estrogen, progesterone, testosterone, but they won't exert their full effect in the absence of thyroxine. So in order to be fully reproductively mature, you have to have thyroxine as well as the reproductive hormones. Synergism is that they do the same thing. So glucagon raises blood sugar levels, but so does epinephrine when you're dealing with acute stress. So will cortisol when you're dealing with chronic stress. And so those would be synergistic, ones that do the same thing. And so when they're both present, then you really get an effect. And then antagonism is where the hormones oppose one another. Insulin and glucagon are opposites. PTH and calcitonin are opposites. ANP and aldosterone are opposites. I'm trying to think of all of our opposites. But when you look at the hormones on the hormone chart, then you should be able to see which ones are antagonistic to each other. Okay, so the last thing that I want to look at here is the relationship between the hypothalamus and the pituitary gland. So in this first picture right here is, here's the hypothalamus, here's the infundibulum that holds the pituitary to the brain. So pituitary is made out of two parts. We have a anterior pituitary, also known as the pars distalis here, and then we have the pars nervosa, or the posterior pituitary. Oh, and the other name for that anterior pituitary is adenohypophysis, means gland, so it's the gland pituitary. This is, oops, sorry, this is known as the neurohypophysis, or the nervous pituitary. So here is what happens with the neurohypophysis. Why do we call it that? Is because we have two sets of neurons that originate in the hypothalamus, and their axons run through the infundibulum, and then their axon terminals end in the posterior pituitary. And in their axon terminals will be secretory vesicles containing these two hormones, oxytocin and ADH. So the posterior pituitary does not produce its own hormones. It just is a storage area for the two hypothalamic hormones, oxytocin and ADH. So yeah, we'll say oxytocin and ADH come from the posterior pituitary, but they are actually made by the cell bodies up here in the paraventricular and supraoptic nucleus of the hypothalamus. So notice in step number 4, when associated hypothalamic neurons fire, so when these guys fire, action potentials arriving at the axon terminals cause oxytocin or ADH to be released into the blood. Now, the anterior pituitary and the hypothalamus have a lot different relationship, because the anterior pituitary does produce its own hormones. It produces these 6 hormones right here. So growth hormone, thyroid stimulating hormone, adrenocorticotropic hormone, follicle stimulating hormone, luteinizing hormone, and prolactin. So GH, TSH, ACTH, FSH, LH, PRL. And I try to use the abbreviations as much as possible. So let's take thyroid stimulating hormone, for example. Let's say that our metabolism is low and we need some more thyroxine. What are we going to do? Well, up here, the hypothalamus is going to be sent a signal from your body that says, hey, you know what? Not making enough ATP, friends. Get me some more ATP. It's probably our muscles going, I need some ATP. So the hypothalamus goes, sure, I can do that. And it will secrete this releasing hormone right here called TRH. TRH will travel down the axon and then be released just like a neurotransmitter into, but it's a hormone, into this capillary bed right here, this primary capillary plexus. So now we have to go down here to the anterior pituitary. So we have 2 veins right here, you know, because after you've had a capillary bed, you have a vein. So I've got these 2 veins called the hypophyseal portal veins, and they will run through the hypothalamus. Then they're going to come to a second capillary bed. And we don't have this much in our body. We have a system like this in our liver, from our digestive tract to our liver, that we'll do when we do blood vessels. But so the hormones will go into, so TRH will go into the hypophyseal portal system, travel through the portal veins, go to the secondary capillary plexus, and then stimulate 4 sets of cells that produce these hormones within the anterior pituitary. All right, so that's how that works. And then that's all the words of that. And then this, I don't even know I have that in there. That's just how growth hormone works. And then here are some problems that can happen with too much, too little, which we'll look at in the hormone chart really quickly in a minute. So too much growth hormone, not enough growth hormone. Here's our thyroid gland. Here are the parathyroid glands on the back. Too much well, in this little boy's case, he's not gotten enough iodine, because in order to make thyroxine, you need to have iodine in your diet. And so his thyroid gland is trying to make, it's being stimulated to make thyroxine, but he can't because he doesn't have iodine in his diet. So his thyroid gland will swell. This person has something called exophthalmos, where the eyeballs bulge out because he or she has Graves' disease. So Graves' disease is an autoimmune disease where antibodies to the thyroid gland stimulate excess production of thyroxine. And so it causes your eyeballs to bulge. You will get a goiter, you'll lose weight, you'll be nervous, and all sorts of things like that. Here's what PTH does, so you can read through that. Here is the adrenal gland. It's got an outside part called the cortex. It has an inside part called the medulla. So these three parts right here will produce aldosterone and cortisol, and then some androgens that you don't need to know. And then the medulla is where the epinephrine comes from, the inside. Here just shows how the stimulus for the adrenal cortex hormones works. And then Cushing syndrome is an excess of cortisol, where you get this redistribution of fat, so you get a buffalo hump on the back, and a lot of other characteristics. Here is the relationship between stress and the adrenal gland. So the secretion of hormones in response to stress. So acute stress or short-term stress is going to secrete the hormones from the medulla, which are epinephrine and norepinephrine. Okay, so sympathetic stimulation. This is going to be more long-term stress here. So we're going to release either or both aldosterone, mineralocorticoids, or glucocorticoids, cortisol, depending on what is the nature of your stress. And so maybe you have low blood volume and low blood pressure, and therefore we need to raise that. That might be the stress in our body. So we need some more aldosterone to do that. Or maybe we just don't have enough ATP to take care of our needs. And so we're putting, instead of glucose in our bloodstream, we're putting proteins and fats due to the presence of cortisol. Here's another thing that you should remember about cortisol is that it is an immune system suppressant. So it's saying, hey, look, I have chronic stress. You don't need to be worrying about using all our energy to make you better or to heal a wound or something like that. We just need you to stop. And so that's why cortisol or cortisone is used to reduce inflammation, because it's suppressing the immune system. This picture just shows the islets of Langerhans that we have within the pancreas. So here is the exocrine pancreatic tissue out here, like it says, and then the cells within the pancreatic islet will produce glucagon. The alpha cells produce glucagon, the beta cells produce insulin. And then this just shows how blood glucose is regulated. If you have high blood glucose, insulin will be secreted. If you have low blood glucose, then glucagon will be secreted. I suggest that you watch these. These videos are actually pretty good about diabetes, and you might be interested in watching that. And then here are the effects of thyroid hormone. They're pretty extensive. And then these hormones are the hormones that are not produced by a gland, but other tissue. When we do the GI tract, we will learn these hormones right here: gastrin, ghrelin, secretin, cholecystokinin, GIP, VIP, might be on another one, nope. And then, so adipose tissue produces a hormone, leptin. The heart produces a hormone called ANP. Kidney produces erythropoietin. Skin produces cholecalciferol or provitamin D3. So that is quick and just, wow, right there to, let's get rid of that. To our endocrine system. So before I leave the endocrine system, let me stop sharing. Does anybody have any quick questions that need to be answered? You can either ask or you can type in a thing. Okay, sweet. All right, let me check my other people. I will get those later. Okay, oh, my videos are, oh, this one, this video I'll post as soon as I'm done. But the other, you know, there's videos already in Canvas so that you can be watching. Let me get my other chat thing. Blanks are on the chart. So if there's nothing in there, then there's nothing you need to know. Yeah, so you mean in the hypo and hypersecretion column? Yeah, no, there's nothing, I didn't need you to know any of that. Yep, so just know the ones that have words in them. Yep, okay. So Gabby, was that your question? Was it this, wherever you are, this video? I'm guessing. I don't know. Oh, there she is right there. Okay, yeah, as soon as this is done. Okay, so let's go then to my blood PowerPoint. Okay. All right. Okay, so here we are in blood. All right, so here are our objectives, fewer than what we had, fortunately, in the last unit. So we just want to talk about what is blood made of, why is it connective tissue, what are the functions of blood, what's plasma like, what are the different kinds of cells like, and then hemostasis. Now don't get that confused with homeostasis. Hemostasis is the ability of your blood to clot itself once a damage has occurred, and then blood typing. All right, so here's just some general information about blood. So we have formed elements and plasma, and I'll show you a picture in just a sec. All right, this number, in fact, I'm even gonna circle it and highlight it and everything. This value right here. Okay, so there are some things that we always want you to know. And in the blood, we need to know the pH of blood. So always and forever, you will know that number. Especially if you become a nurse and you're having to do ABGs, you have got to know arterial blood gases and acid-base balance. 7.35 to 7.45 is the homeostatic range for blood. And then notice that's the homeostatic range, but here is right where we want to be, right smack dab in the middle. So 7.4 is the pH of blood. Our amount of blood that we have, you'll want to remember this number right here, especially when we get into cardiovascular system. We'll talk a lot about this with cardiac output. So 5.25 liters of blood is about what everybody has. I probably don't, because I'm a little person, and so I'm probably down here in the 4 range of blood. And then bigger people may have maybe even a little bit more than 6 liters of blood. Okay, here's another bunch of numbers that you'll want to know is hematocrit. So hematocrit is the percentage of red blood cells that you have in a volume of blood. It doesn't matter how much blood you're taking out, if you're taking out 500 milliliters of blood or you're just taking out a capillary tube of blood, whatever, it should always be right around 45% of the whole blood should be red blood cells. Males, you'll notice, 45 or 47, sorry, plus or minus 5 would be the percentage in a male, and females 42 plus or minus 5. So we can be anywhere in those ranges. Why do males have more red blood cells than females? Because they need more oxygen. So they'll be carrying, they'll have more red blood cells to carry more oxygen to their tissues because their metabolic rates are higher than in females. All right, so here shows a column of blood once we've spun it down. The heavier material, of course, is gonna bullet down here to the bottom, so we'll have the red blood cells down here. Then we have this buffy coat, which is also cellular in nature, and it's made up of the white blood cells or the leukocytes and the platelets. Okay, so, and it's gonna be about well, less than 1% of blood, because obviously 55% of whole blood is the liquid portion, or the solution, or the solvent portion. I have to stop and think what that's called. The solvent portion of blood. And so it's going to be, we want it to be a majority of the blood, because that's what allows the blood to flow. If we were to increase this, so in other words, if we were to shift the percentages so that my red blood cell level went up to here, let's say I have polycythemia, I'm an athlete, and I'm working out up in the mountains, and I'm producing more red blood cells. So now if I'm up here to maybe, well, I don't know, we can have a hematocrit of 60 in some athletes. Wow, that means I only have 40% plasma. Is my blood going to be thick and sludgy? Yeah, it is. But you probably have a super good heart that can help propel that blood through that cardiovascular system. And so that's what's going to happen. We're not going to change this level at all. Okay, that's going to be always the same. What we're going to do with hematocrit is just change where this line is. It's going to go up, it's going to go down. And so then, of course, this line would go down. Okay, so that's how hematocrit works. All right. Okay, now why is blood a connective tissue? Because embryonically, blood comes from a stem cells called hemocytoblasts that originated from, where is my word? Mesenchyme. I don't know if you're familiar with mesenchyme, how much your instructor from 201 talked about it, but I talk about it a lot. So I like to talk about embryonically, how do things form. And so the hematopoietic tissue, or our stem cells, our hematopoietic stem cells that we find in the bone marrow, are part of the cell line from mesenchyme. Mesenchyme can become 4 different types of tissues and connective tissues, and so that's why it's a connective tissue. Okay, what are the functions of blood? They distribute things, nutrients, wastes, hormones, whatever need to be carried throughout the body from inside to outside and from place to place. The blood is going to do it. The second thing is regulation. So we're going to be regulating body temperature, pH, fluid volumes, nutrient levels, I don't know, whatever. And so that's a very important aspect of blood, and we'll be talking about that a lot this semester. And then the protective function of blood is to prevent blood loss. And so by having the platelets and the clotting proteins, we can have hemostasis, or we prevent and combat infection through the white blood cells. All right, what is plasma made out of? You can look, there's a chart that shows the different substances within plasma. There's some more of them. Here's the one thing that I got to have you know, so that we can always talk about things later on, about this concept later on, and this is albumin. I don't know if you're familiar with this concept right here of colloid osmotic pressure. I don't know if you've talked about that in 201 before. I know that when we talk about cellular transport processes, that I make sure my students know that. But this is the tendency for water to flow to a more concentrated area. So in order to get water to move somewhere, you got to make it want to go somewhere. And so if we want water to stay in our blood, one of the things is that when we go down to the capillary level, water is going to be pushed out into our tissues, which is fine. We need that to happen in order to take the nutrients and everything that's inside of our plasma to our body cells. But we don't want that fluid to stay out there, because if fluid stays out in our interstitial fluid, or stays out in our tissues, then we get edema and we'll get swelling. So to stop this swelling from happening, we need some plasma proteins that will not leave the capillary. So they'll stay within the capillary blood, and then they will draw the water back in. After the water is forced out, then the water will want to come back in because of osmotic pressure. So albumin is a molecule made by the liver, most abundant plasma protein to maintain osmotic pressure of the blood. I don't care that you totally get that concept right now. When we do capillary dynamics and then when we do filtration, again, with the urinary system, you absolutely need to know that. So it might be nice between now and then if you're just, you know, somewhere and you've got 5 minutes and, you know, pull out your phone and look up osmotic pressure and make sure that you have, you know, some kind of command of that before we get to it for reals. The plasma proteins can be shuttles, buffers, clotting proteins, and for immunity, like the antibodies. All right, there's that. Then the formed elements are the cellular part of the blood. Here is the amount of red blood cells that we have, about 5 million. Homeostatic range between 4 and 6 million per microliter. So that's 1,000th of a milliliter. That's super tiny, or 1,000,000th of a liter. So that's, you know, very, very small amount of blood, but tons and tons of red blood cells. Red blood cells, as you know, are biconcave discs. So they're not spheres. They're squished in the middle. They don't have a nucleus. They are very flexible to travel through a capillary because a capillary is as wide as a red blood cell. So the red blood cells, as they pass through your bloodstream, go in single file line so that the O2 that is carried by the red blood cell can jump off and into the tissues, and then the CO2 that is in the tissue, some of it can go into the red blood cell. Most of, well, it goes into the red blood cell for part of the time and then it leaves as bicarbonate, but we'll get to that with the respiratory system. All right, why does it need to be a biconcave disc? So that we can have a huge surface area to volume ratio for gas exchange. Now, this is just a really important concept too. So yeah, if we're gonna meet on next week, then I don't have to hurry. We can take a little bit. We'll do hemostasis next week and then we'll be good. Because I really wanna show you how oxygen works in a red blood cell. Now, let's say that we have a red blood cell that is a sphere. Okay, so we're looking at, this is the side view of my sphere. Okay, now notice that it's dipped in on both sides. Okay, just like that. So why do we want that? Couldn't I carry more hemoglobin molecules in a bucket like this than in a bucket that's shaped like that? Well, it's not the question of how much hemoglobin can I carry, but it's how much O2 can I exchange. So let's say that I have a hemoglobin. Remember that if you want the O2 to pop off, the hemoglobin molecule, so that's what I'm drawing, these little blue things are hemoglobin molecules, they have to touch the edge so that the O2 can jump off. Okay, I don't know how many of you have been to England before or familiar with the Underground, but there is a subway in London called the Underground. And when the little trains, so here's the platform where you're standing, and you're gonna think, well, you're stupid, lady. No, this is really important. And then there's a gap, and then here's the train. Okay, so there's a lady that, a recording woman that has narrated or, you know, announced for the Underground for a long time. Then they fired her, and I don't know if they hired her back, but anyway, it was a real big thing because people were really sad. So you're standing here on the platform and you want to get into your train. So you want to have the doors open. You want to have as much space as you can get in so that you can jump the gap and get into the window. Get in through the door. And then what if your stop is really close? Well, you want to stand right here. You don't want to stand in the middle where you can't, like out here and out here, where you can't get off the train when your next stop comes. And so ideally, a train should have lots of openings and be really long and skinny so that everybody can be close to a door so you can get out. Well, that's what the red blood cells want. So the red blood cells want their hemoglobin right next to the edge so that you can jump off when your stop comes. Now, I don't know how many of you are familiar with Divergent, the book or the movie, either one. But remember, for those of you that know it, well, I'll just tell you. So there were these kids that were in these different groups. kind of like being sorted in Hogwarts, but worse. And so one of the groups were the Dauntless group. And one of the things you had to do in order to become a good, like really sell yourself in Dauntless, was that there was these trains that came by that never ever stopped, just like red blood cells. So what do you have to do to get across onto the train that never stops? You have to jump. So you want a big opening in your train, and you want to be right here on the edge, and you don't want a gap. You don't want very much space in here. What if I put red hemoglobin molecules inside of here? Could the oxygen ever get off? Probably not, because it's so far from the edge of the red blood cell that the diffusion of O2 out of the out of the cell in your tissues and into the cell in the lungs would not be efficient. And that's why you want to be a biconcave disc. I love them. You guys, you don't even know. There are some things in your body that I think are absolutely amazing, and I just love it. Okay, so hemoglobin will bind easily and reversibly, which is super good because you don't want it to bind to not bind, but you want it to not stay bound. You want it to pop off when you get out to your tissues, but you want it to bind really, really tightly in your lungs and not so tightly in your tissues. So every hemoglobin molecule will carry 4 molecules of oxygen. This is your homeostatic range of hemoglobin in your blood. If we look at this picture, we see this is a hemoglobin molecule. There are 4 globin chains. They're all folded up on themselves. And then we have a heme molecule that has an iron atom in the center. And then O2 will bind to the irons, creating iron oxide. So that's why your blood's red, because rest is red, right? So anytime oxygen and iron come together, they make a red color. And so that's why it's that. All right, so those are the 4 each hemoglobin. There's about 250 million hemoglobin molecules per red blood cell. And then if you have if you have 5 million per microliter and you have 5.25 liters of blood, we figured out that that's upwards of 10 to the 26th oxygen molecules that can be carried by your bloodstream at any one time. So that's a big number of oxygen in your body. All right, so moving on, we have a process called hematopoiesis, which is the production of blood, and specifically blood, hematopoiesis. But then if we want to make just red blood cells, then we have a process called erythropoiesis. Happens in the bone marrow, comes from the stem cells called hemocytoblasts right here. They're going to go through this process to be turned into a red blood cell. In order to go through this pathway, you need 3 things. You need iron for the hemoglobin. You need vitamin B12 so that you will go from this reticulocyte, well, this is a reticulocyte, into a red blood cell, or even this macrocyte into a microcyte. And then you need folic acid as well to form the cell properly. So without these things, then you can have different types of anemia, pernicious anemia and iron deficiency anemia. All right, red blood cells, because they are anucleate, do not live very long. They live about 120 days. Once they become damaged on their pathway through the body, their cell membranes become very ragged, and they become trapped in the spleen. So the spleen has these big sinuses of blood inside of them, and they have cells inside of them that are checking for old red blood cells. So then they'll get stuck and removed from circulation, and then new ones will replace them from the bone marrow. So you can think of the spleen as the red blood cell graveyard, because that's where they're going to end up. We're going to take out the iron and store it, and then the rest of the heme molecule is going to be turned to bilirubin and then bile, and then go into our intestines, and then we'll defecate it out. And that's why feces look the color that they do, is because it's the breakdown product of red blood cells. When your liver doesn't function properly, as in cirrhosis of the liver or hepatitis, that bilirubin can't be processed, and then you have jaundice. And so you look yellow because, you know, if you've looked at a bruise before, remember if you got that purpley blue bruise and then it's going to turn green and then it's going to turn yellow. Well, it's going to turn brown, then it's going to turn green, then it's going to turn yellow. Those are the different products of the hemoglobin being broken down, the different colors it goes through. Okay, erythropoietin is that hormone that comes from the kidneys that stimulates red blood cell production if there is hypoxia, so lack of O2 in your tissues, especially your kidneys. All right, I want to get to the hormone chart. So I'm going to stop there. Find my stop thing. And then, because I want to make sure that you understand. Oh yeah, I heard about that, Peyton. So Peyton just said the lab book is sold out in the Glendale bookstore. So it's this one. If you don't have that, I will, let's see. So what did I put? It's not so worrisome that you don't have it until midterm, lab practical time. So when is our lab practical? Is June the 18th. Oh yeah. So because I don't think I used any of the pictures from the lab manual on the first quiz. So I will, the ones that I used on the quiz, I'll make sure that I upload into Canvas. I'll just scan them and upload it into Canvas. So let me know if people that don't have the lab manual, at least by next week. But yeah, I did hear that they didn't have any. So let me make a note. I'll check with the bookstore and find out when they're supposed to be getting them. I got an email last week. I think there was a problem with the people that publish it, these Blue Door people, you know, they're kind of not a big, huge company, and so it takes them a while to do things. So I think it'll be forthcoming, but I will check on that today. Okay, so like I said, so anything that we need, any pictures that you need for the quizzes, I will make sure that you have. All right, let's look at the hormone chart and show you. I did a couple of things. Yeah, it's the same lab manual. So Corey asked, yes, let me go back here. I got two more questions. So we have quick question, is plasma oncotic pressure the same thing as colloid osmotic pressure? It is exactly the same thing. I don't know why we use two different terms. And the frustrating thing, I know, I try to use just this colloid osmotic pressure all the time. I have a graduate renal physiology class that I'm taking right now, and my professor in that class, she just switches back and forth. Half the time she calls it osmotic pressure, half the time she calls it oncotic. I just know that they're the same thing. So yeah, exactly the same thing, because they do both pull water, and I don't know why we need two different terms. And then, is the lab manual for this class the same as the one from 201? Yeah, that's exactly the same thing. Okay, so okay. All right, so in the last few minutes, let me show you some things to focus on. So this is, Rihanna asked about this. Oh, here we go. The blank spaces. Yeah, there's nothing that I need you to know in here. This is an older chart. Yours might not look exactly like this. I just realized it's not exactly the same one, but it's got the same information, so you're going to be fine. Reason why I know is because I took out progeria, so you don't need to worry about progeria. We're not sure that lack of growth hormone is the only thing that causes progeria, but definitely dwarfism. But when you think about it, if you know what progeria is, it's that where the little kids age really super fast, so they look like, you know, 80-year-old people by the time they're like 6. You know, it's a form of pituitary dwarfism too. So anyway, but there are other things that cause progeria besides that. Okay, so down here, I hope I put those. See, you guys, I didn't. That's why I don't like this chart. If you look at your chart that's in Canvas, it has the key for these letters and numbers. Okay, the numbers are the processes that are back in our notes: reproduction, growth and development, mobilizing body defenses against stressors, fluid, electrolyte, and nutrient balance, and what was 5? Metabolism. So that's what the 1, 2, 3, 4, 5 stands for. Okay, then B and D and E, A, B, C, D, E are the things that the cells, so target cell activity. So alters membrane permeability. Oh, am I going to remember what they are? Alters membrane permeability. The first tropic one stimulates the production of another hormone. C is activates and deactivates enzymes. D is secretes the hormones, and E is mitosis. So I just put those in there to help you understand what these hormones were doing. So what I need you to do for the chart, number 1, is know the glands and what hormones come from them. Okay, that was part of in the quiz to really reinforce what you need to know. So glands and the hormones that come from them. Okay, then to go even further, we need to know what are their target tissues. Okay, so we can see all of the releasing hormones from the hypothalamus go to the anterior pituitary. Where does growth hormone go? Liver, muscle, bone, cartilage, other tissues, adipose. Where does TSH go? Thyroid gland, and so on. Then when they get there, what do? Okay, stimulate growth, stimulate release of thyroid hormone, stimulate sperm production, whatever, promotes lactation. Now, in some cases, there will be a disease that you get from too little. That would be a hyposecretion of the hormone, or too much is a hypersecretion of the hormone. Okay, so the down arrow is a hyposecretion, the up arrow is a hypersecretion. So you should be fairly familiar with these. Let's look at the cretinism and Graves' disease. Let's look at it in thyroid hormone, because it's also there with thyroid hormone. So if we come down in the chart, if I have too little thyroid hormone, this is hypothyroidism. In a fetus or in an infant is called cretinism. Hypothyroidism in adults called myxedema. Usually we just call it hypothyroid. You know, you talk about, oh, I'm just tired and sluggish and I'm gaining weight and I'm cold all the time. Maybe I have, my thyroid isn't functioning, hypothyroidism. And then hyperthyroidism is this autoimmune disease called Graves' disease, which gives you goiter and exophthalmos. Exophthalmos is that bulgy eyeball thing. And then Addison's disease, Cushing's, from too much, too little, adrenal cortex hormones, and then pancreas, you're familiar with those. Now, notice I didn't put anything in with the testes and ovaries. I just listed the hormones because we have an entire chapter on that at the end. So you don't need to remember anything, just that the testes produce testosterone, and the ovaries produce estrogen and progesterone. Okay, now the last thing I want to look at is just a reinforcement of these other hormone producing structures. So the heart, it produces ANP, or atrial natriuretic peptide. It goes to the kidneys, and its purpose is to decrease sodium uptake. So we're going to excrete sodium, because there's a saying that goes with sodium in your body, is that water always follows salt. So if you have too much congestion in your heart, too much blood, basically too much blood volume, then if we excrete sodium, then water will go with it. Water will stay in our urine and our blood volume will go down. Okay, so ANP, that's its job, is to decrease sodium uptake, and then consequently is to lower blood pressure. And we'll talk about that job when we do blood pressure. The placenta makes human chorionic gonadotropin, HCG, goes to corpus luteum to help maintain pregnancy. Kidneys produce erythropoietin, goes to the red bone marrow for red blood cell production. The skin produces cholecalciferol that goes to the kidney, bone, and small intestine, but its main job is to go to the small intestine so that you can absorb calcium. Because you know what? If you don't have vitamin D, you're not going to absorb calcium from your small intestine. You could eat all of the calcium that you want, drink it in your milk or eat in cheese or whatever, and you wouldn't absorb it. And then lastly, adipose produces leptin that goes basically to your hypothalamus to tell you, for satiety, to say, hey, look, Stop eating. You've eaten enough. You're full. All right. Woo. So I know that's a whole bunch of stuff. And I really feel bad, but at least you get this. You get me live sometimes, but I hope that you've been able to get into the recorded lectures that I've done and watch those and everything. So let me double check. Let me look at my participant. Looks like everybody's here. So I got Alyssa, I got Ashley, I got Brianna and Shell, I got you, and whoops, Corey, I just lost you off my list. Corey and Daniela, got you, and Elizabeth I got, and Gabby I got, and Jacqueline and Julia I got, and Leslie got you, and Mariah and Martina and Nicole and Peyton I got, and Savannah I got, and Susan, Tina, Warren, and oh, there's Willem and Jacqueline. Okay, so okay, all right, 1, 2, 3. Okay, perfect. All right, so before Zooms are an hour and 15 minutes. Oh, okay, so Alyssa, oh yeah, Alyssa, yeah, oh Haley, I didn't see you. Sorry, where are you on my list? I'm glad you just clicked on my list, sent me a message. Yeah, I don't even have you. Oh, there you are. Sorry. Haley, you're here. Thanks for that. Sorry about that. Yeah, because I didn't know how to pronounce your first name, so I just looked at Tano. Tano, I don't know. Anyway, I saw you. You're good. Tino. Okay. Tino. All right. That's because I thought, I looked at that, I was like, I am gonna butcher that. So I was embarrassed. I didn't want to say your name wrong. And oh, Samantha, I didn't see, that was somebody else I didn't see. Samantha, where are you? Samantha Librado, right? Is that okay? Yes. Excellent. I got you. Yeah, I don't know why you didn't show up on my list. Okay. Tino and Samantha and extra lab manual. Okay. So Corey has an extra lab manual. Who needed the other lab manual? Where are you? Oh, Peyton. So if you guys, so Peyton Castaneda and Corey Lowen, if you guys want to hook up in the inbox, Corey has an extra lab manual. And okay, perfect. Oh, and Haley Bove needs one too, if anybody needs one. Okay, here are, okay, so Nicole, the class is scheduled to meet for an hour and 15 minutes. So we just barely got over that. So 9 to 10:15 is what we'll meet all the time, except for next Tuesday. Okay, so while we're here, what do we want to do next week? Do we want to meet on Monday? Anybody like, okay, and here's a thing about next week. I will record it and I won't take attendance. I'll just, you know, everybody gets a free pass on that. So if you can't make it, that's cool. We'll be fine. And then you can watch it. I'm gonna have to let you know what time that is. But you'll, just like you got that message from me yesterday in the inbox with this time on it, it'll be a different Zoom number because they'll give me, well, I'll probably use my personal ID or my personal meeting room that I have with another school that I teach for. But this one I've scheduled in Zoom for the rest of the semester. So it'll always be the Zoom ID that you signed in with today, okay? But Mondays will be different. So I will be sending you one probably Sunday once I figure out what my life's gonna be like once I get to North Carolina. And if I have internet, I may have to go try to find some place for internet. Let's tentatively schedule it for, let's see, my time, so that'll be 3 hours later. So Monday afternoon, how about noon on Monday? So let's go ahead and say noon on Monday. That would be 3 o'clock for me. And if I have to push that back further, then I will. So my 3 p.m. Eastern. So that's noon for you guys. Okay. All right. Okay. Yeah, so that's Alyssa just said check OfferUp, Craigslist, Facebook Marketplace, and Amazon for lab manuals or book. Haley, so you guys that have here, let's do this. If you have questions as you've been studying in the blood stuff, or even anything, I don't care, just send me a message in the inbox in Canvas, and I will address that in the meeting, and then hopefully, and I'll just get it posted right after it's over with, and then hopefully you can see that before you take the test. Oh, the test's on Tuesday, so yeah, you can watch it like Monday night when you get off class or work, or hopefully. So does that work for Haley and Nicole? Yes, and I will well, we can discuss right now. So totally, yes, Gabby said, could you possibly do an open discussion for any questions? That's what I hope. That's most of what I want to do on next week, is to go over anything that you're stuck on. Okay, so yes from Haley. So hopefully, Nicole, you'll be able to watch it before you take the test. Okay, sweet. There you are, nodding your cute face. Okay. All right. Okay. So if there's nothing else, then I guess that's it. So I am going to finish. This has to, whatever it does thing in Zoom, and then I'll post it. It'll take me, because it's long, it'll probably take about 20 minutes to get it posted, but then it'll be be in, let's post it in the announcements. Okay. So just the top announcement that you get will be this. Whatever you want. Nicole just said, will labs be done on Zoom too? Probably not. But if you have any questions, then yeah. So when we're doing like the lab practice, because the quizzes are like 10 points. They're not a ton of things to worry about, but the lab practical is 100 points. Go through the lists that I gave you. So I gave you a list. Let's see, where's my list? So what's on it? So blood and endocrine glands, heart anatomy and physiology, and the blood vessels. So those first 3 lists, then print those off, and then just go through all of that and make sure that, mostly because it's mostly identifying, but then there are some questions next week that we'll talk about. Also, we'll talk a little bit about the lab on heart anatomy and physiology and blood vessels too. So yeah, we'll just hit everything. Well, whatever we can fit into in an hour and 15 minutes. I didn't make a video on the blood vessel lab because you just have to know the pictures, just where they are. So there's probably nothing I can teach you about that. But I did show, when you look at the heart anatomy and physiology videos, there's good stuff in there. So, okay. Alyssa just said an open discussion board would be nice. I can make one of those. I don't have any discussion board. Let me go to our class. And sure, I can make a discussion board. So I will do that. I'll add that for each. Oh, so what I'll do here, let me show my screen so you see what I'm talking about. because you guys don't have, see how I have a little you can't see it thing? So I will make that visible, and then I'll move it up into here where you guys can see it. And so then we will have discussions on each topic. So I'll make one for endocrine system, I'll make one for blood, one for heart, one for everything that we do. So there will be that too. Okay. And anything else? Five, four, three, two, one. Okay. Okay. All right. So have a great day, week, whatever, till I see you again. So I gotta, okay. So Monday at noon, Monday, June the 8th. Okay. And then remember, be looking for a different thing, a different Zoom ID than what we used here. And thank you. Oh my gosh, you guys, so gross. I don't know if you've ever had to drive to the East Coast before, but wow. So anyway, I'm not looking forward to that at all, but you know, you got to do what you got to do. All right. So it was good to be with you guys, and I hope that I can help you with whatever that you have, and that you enjoy the class, and always contact me with anything, situation, stuff that you have. And there it is. Oh, discussions. I will post those while I am waiting for that to upload. Okay, great. Have a fabulous day. We'll see you. Bye.